So, well, thank you for joining us again for the third week of the short course. This is, at least for me personally, perhaps one of the more interesting aspects of the short course. And going into different aspects of the technology on the first week and the second week, the aspect of technology that I particularly find interesting is the identification of metabolites. And like the last 2 weeks, we're going to provide some of the real basics behind how we do these activity metabolomics experiments. And then we're going to delve into some of the more, I think, more challenging or more sophisticated aspects of this, which the one that this week I'm particularly interested in. So metabolite identification and lipid identification is still considered one of the major bottlenecks in metabolomics. And I think it is the reason why you really can't put metabolomics into the area of digital biology. I mean, clearly genomics, transcriptomics, proteomics, you can easily put those fields into digital biology. They're being applied and routinely being used to expand upon the field of digital biology. However, metabolomics still suffers from the aspect of not being able to get solid identifications on these molecules. And as a result, it's really hard for us to make that jump into what is now covered by these other fields. So today I'm hoping to, on some level, address this area of digital biology. And I say digital biology, I wish I had thought of this concept with respect to metabolomics, but it was really pointed out to me about a year ago by Martin Guerra, who is a colleague and a friend of mine. We co-authored a number of papers together, and the last paper I'm going to talk about today is going to cover this specific topic. So we're going to have 3 speakers today. Michael Kersey from AstraZeneca, and also he's a professor at University of Gothenburg in Sweden. He's going to be talking about the application of imaging, and it's a very unique application of imaging. He's one of the few people in the world who actually can do this, and that's subcellular imaging with absolute quantification. So he's doing organelle level imaging and also accomplishing absolute quantification. And he's one of the individuals who actually developed this technology and has made it, I wouldn't say mainstream, it's still a relatively small handful of individuals who can do this, but I think Mike's relatively short presentation will be equally as inversely proportional to how short it is, it'll be equally as powerful. The other 2 speakers, who are extremely reluctant speakers for this, is Lin Huang, who I'm pretty sure now I've had more papers with Lin than anybody else. She's been a major backbone of metabolomics in our lab. The postdocs tend to go to her. And because the data she creates is so reliable, and everything she does is very carefully done. So it's really nice. And I think this is the first time I've ever seen her give a presentation. So thanks, Lynn. And also Winnie, actually, so I'd like to say Lynn has had a huge impact on the metabolomics community, which she has, but Winnie, I think, probably is even more impactful in that respect, in that she has basically created the Metlin database, or at least 98% of the Metlin database has been generated because of Winnie's efforts, which is well over 910,000 molecules are the result of Winnie generating that data. So you're going to hear from both of them in their firsthand experience in generating the database and also doing metabolomic type of experiments. Like last week, I did mention that the book is still, and it will always be freely available. You can get it as a PDF at this particular website. And it's also available on Amazon at a royalty-free price of $13 if you're interested, but there's no reason to buy it. Just download it free. The whole thing is right there. And just quickly, I'll mention the future speakers. I've already mentioned Martin Guerra from University of Leiden or Leiden University Medical Center. He's going to be talking next week at activity metabolomics specifically, and looking at endogenous molecules and taking those to the clinic. This is a decades-long effort on Martin's part, so I think this is going to be a really interesting talk for everybody who goes to it. And I'm also really pleased that we've added another cameo speaker. Her name is Shua Han from Duke University, and her specific interest is in the gut microbiome and identifying bioactive metabolites from the gut microbiome. So it's both speakers will have something interesting to say. And of course, I'll be going through some of the things that we've been doing in activity metabolomics as well. And I may bring in a third speaker, but I haven't quite decided on whether we'll be doing that or not. I'm still working through that one. And with that, I've already shown you the script, the image of Scripps, and also this is the room that we're currently located in. Coming to you from Scripps. This is my lab, and as I already mentioned, Winnie Urabuntai / Hein is going to be her, and also Lin Wang will be talking today. And the cameo will be Mike Kersey on subcellular quantitative imaging of organelles. And today it'll be kind of a combination of different things, including untargeted versus targeted analysis, the XCMS in untargeted analyses, the creation of the Metlin database, metabolite identification, and also ultimately the integration of XCMS Metlin. And the primary focus of this, as I mentioned at the very beginning, is how we're trying to get metabolomics into digital biology, really make it a technology that can truly integrate in with genomics, proteomics, and transcriptomics. So last week, Elizabeth and Bill talked about our targeted metabolomics-based approaches on a fairly basic level, giving you an idea of how we do these experiments with the triple quad. And then Liana further developed that concept on a much grander scale with her population scale metabolomics and her ability to look at hundreds, if not over 1,000 molecules at one time in a targeted sort of way. Today, we're going to focus in on global or untargeted metabolomics. I particularly like this area from an activity metabolomics perspective because activity metabolomics, we're ultimately trying to identify molecules that have specific biological activities. And you can't always predict, as has been shown in multiple occasions in our lab, you can't always predict what molecules are going to be active. So the nature of a targeted-based approach is that you pick molecules you're going to look at, and you look for up or down regulation, and then try to make decisions as to which molecules are the most interesting from a biological perspective, and you start testing them. From a global perspective, we don't make any of those assumptions, and we let the biology tell us what's actually happening. And we've actually had a number of very nice successes, and Lynn is going to describe one of those, and I'm going to expand upon that a little later on. And the technology we're typically using for untargeted analyses are quadruple time of flight mass spectrometers in our lab. Also, the Orbitraps are commonly used for this as well. We have a Q Exactive that we occasionally use for these sorts of analyses. And from a metabolomics, I always like to bring in a historical perspective. I think one of the first papers that actually described doing this sort of analyses was back in 1966 by Dick Leash. This was also a paper that was pointed out to me by one of my former postdocs, Bill Wyckoff. I'm not sure if I ever would have found this, but it's really an interesting paper that basically describes looking at as many molecules as you possibly can stimulating those molecules within a biological system, or stimulating the system and using the technology to identify what those molecules are, and ultimately then testing them for their biological activity. And ultimately, it is the more sophisticated nature of the global metabolomics-based approaches are result in quite a few more steps than in a targeted-based approach. Fortunately for us, it generally hasn't been a major bottleneck. It just requires a little bit more effort in designing the experiments and also preparing samples. And with that, so basically you have the pre-analytical based approaches, which are, you have the samples and the quenching of the metabolism and the storage, the analytical based approaches, which include extraction and the actual data analysis, and ultimately the post-analytical based approaches, which we've focused on with respect to AMMS. And Lynn Wang is going to actually discuss that in a little bit more detail right now. And then later, we're going to have Winnie discussing Metlin. So thank you, Lynn.
Thank you, Gary. Hi, everyone. My session today is on global metabolomics. For this process, I first start with its workflow and then move on to a specific global metabolomics analysis that I had done in the past. The first step of workflow is sample preparation. Sample must contain 20 to 100 microliter of fluid, 10 milligrams of tissue, and at least 1 million cells. However, it depends on the metabolite that you are interested in. Chloroform methanol 221 is used to extract lipid classes, while methanol acetonitrile water 221 are only methanol can be used to extract polar metabolites. Normalization of sample is done by the sample weight for tissue or the sample protein contents for cell before LC-MS analysis. We have our standardized instruction protocol that are posted on the Metlin website. The next step is data acquisition or LC-MS analysis. The extracted sample are analyzed on UBLC coupled to a Q-top. For non-polar metabolites, the LC-MS analysis is performed by the reverse phase in both positive and negative ion modes. For polar metabolites, LC-MS analysis is performed by helix in both positive and negative ion modes. The collection of both MS and MS2 data occurs at the same time. After the acquisition is done, the MS data is uploaded to AMMS to run single group, pairwise, or multiple group comparison. AMMS perform big detection, retention time correction, big alignment, big iteration, statistics, and annotation. AMMS detects 20,000 to 40,000 features. The next step is to prioritize significant features. We use statistical p-value, fold change, and intensity cutoff. From this, we typically obtain hundreds to 1,000 significant features. The last step is searching the collected MS2 data against Metlin database to identify the significant features. Now I am presenting to you a global metabolomic analysis that were performed on plasma sample of mice infected with a virus, LCMV clone 13. There are two mouse strains, DBA1 and DBA2. DBA1 has cytokine storm, and the DBA1 mice die after 1 week of infection. The mice die not from the infection, but from immune response. The DBA2 had T-cell exhaustion, and the DBA2 mice survived from the infection. Plasma samples were collected before virus inoculation, day 0, and after 5 days of infection. There were 4 groups of samples and 10 replicates per group. Metabolites were extracted from plasma by adding 400 microliter of whole methanol to 100 microliter of plasma. LCMS analysis was performed by the reverse phase in positive mode. The MS data were uploaded to XCMS to run pairwise and multiple group comparison. XCMS detected about 23,000 features for pairwise comparison and 28 features for multiple group comparisons. After using the cutoff with a p-value less than 0, after using the cutoff with p-value less than or equal to 0.01 and mass intensity greater or equal to 30,000, there were 289 significant features obtained for pairwise and 783 significant features obtained for multiple groups. Here is a significant dysregulated feature from pairwise comparison, DBA1 day 5 and DBA2 day 5. This feature had MOG 190 at retention time 21 minutes and had p-value 7 times 10 to the power of -6 and had good fold chain of 16. And here is another significant dysregulated feature from multiple group comparison of 4 sample groups. And this feature had MOG 205 at retention time 4.9 minutes and have p-value 1.3 times 10 to the power of -6. To identify the significant dysregulated feature, MS data were collected from the pool sample and was searched against the Maclin database. And here is the Maclin hit for the feature 190 MOG. And here is the Maclin hit for feature MOG 205. In the Maclin hit for feature MOG 190, the top spectrum is the Maclin MS2 of 3-indole-probiotic acid. And the bottom spectrum is the experimental MS2 of MOG 190. In the Maclin hit of the feature MOG 205, the top spectrum is the Maclin MS2 of tryptophan, and the bottom is the experimental MS2 of MOG 205. In conclusion, the two metabolites identified were down-regulated in the infected mice, but upregulated in the infected mice that survived from the infection.
Yeah, so what Lynn just described, it sounds like a fairly simple experiment, and it is. I mean, it's relatively straightforward, and of course, there are many other molecules that she observed in these experiments. But she focused on these two molecules. One is that tryptophan is one of the precursors to generating the indole propionic acid. And indole propionic acid is a really interesting molecule. And I could almost say it's probably one of the more interesting molecules in my 34 years at Scripps, almost 35 years now, that we've identified. This molecule was observed originally in a set of experiments that we did with Pete, in collaboration with Pete Schultz. Actually, Pete initiated this whole study when he was at the Genomics Institute of Novartis. And it was a really, to me, it was a really insightful approach to doing metabolomics in a way that had been extremely novel at the time. So the proposal here was to actually identify molecules associated with animals that didn't have gut microbiome and those associated that did have a gut microbiome. So it was comparative analysis between the two. What was interesting about the indole propionic acid was that it exclusively exists in the plasma of the animals with a gut microbiome and did not exist at all in the animals without a gut microbiome. And ultimately, through extensive efforts by Bill Wyckoff and Eric Peters, if you can see that they're co-authors on this paper, they ultimately were able to identify the specific bacteria that was producing this. So academically, that's really interesting. But what's even more interesting has been the biological activities that's associated with this particular molecule. On this particular paper, we've had over 2,800 citations on it. And I believe a lot of those citations, based on the ones that I've looked, are coming largely because of the observation of this particular molecule. And I'll show you the study that Lynn just mentioned. That particular study showed the immunosuppressive effects of indole propionic acid. So the animals that had been infected with the virus and survived are the animals that had a higher level of the indole propionic acid. And as Lynn said, the animals weren't dying because of the virus itself. They were dying because of their immune response to the virus. So the animals that had a lower level of indole propionic acid didn't survive, whereas the ones that had a higher level did survive. And this has been the immunosuppressive effects of the indole propionic acid has been documented in many cases through those publications that I mentioned earlier. But it's also had a variety of other implications in both cancer and also this very recent Nature paper where they name it out specifically where it promotes nerve regeneration and repair. So from a metabolomics perspective and also from an activity metabolomics perspective, I think this is a great example of what can be discovered using untargeted metabolomics or global metabolomics. So I mentioned, one of the key aspects to this has been the Metlin database. Metlin is a fairly extensive resource right now, and it started off in the early 2000s to facilitate molecular identification. And it started off where we just were putting in molecules into the database and using the elemental composition or the MOZ values to help identify what these molecules are. As you can imagine, that is problematic because a lot of molecules have the same elemental composition, or even if they have different elemental compositions, their masses can be very similar. So the redundancy with respect to using Metlin in this way was with respect to different molecules showing up was too high. So we needed to add another component to Metlin And that was where we started generating MSMS data on these individual molecules that we accumulated. And that MSMS data added a significant addition to the level of characterization that typically allows us to do fairly good identifications on each of these molecules. Here's a couple of examples of caffeine, simple MSMS spectrum for those of you who have not observed this sort of data in your experiences. And this is actually a molecule that we analyzed back in 1994. This was with Richard Lerner and Ben Crabat. This was a Leamide, and this was in a sleep study that we were performing on cerebrospinal fluid. And this is the molecule that we identified as that was associated with sleep deprivation. And it turned out to be a science paper at the time. So we began with roughly 100 molecular standards. And since 2017, it's really undergone significant growth. So as of roughly 2020 or so, we were up, or 2021, we were up to about 850,000. Now we are at 935,565. Is that the accurate number, Winnie?
Yeah.
Okay. It might be 300, it may be 935,566. I'm not sure. So this is where Winnie's going to give you a little bit of her own personal history behind this, and then I'm going to go back into the whole digital biology space.
Hi everyone, this is Gary. So a little bit history of Medlin. At the beginning, Ted Brendan, Johnny Pond, and Sean Quinn had added Medlin entry manually one by one. And then in 2006, Diana Wong and Paul Benton has joined the lab. And this time they don't have to add entry manually anymore. They left for grad school later in 2008. And that's when Kevin Shaw and I has joined the lab. Has taken over. And Kevin improved Medlin even more. As you can see, we were pretty young, and especially Kevin. And then Kevin then left for grad school. And Ari Espona has joined our lab, and he has done greatly for Medlin and our lab. And now we have roughly 935,000, over 935,000, compound with MS data. With that big number, where did we get our compound? Our compound mostly donated by many vendors such as Avanti, Cayman, Sigma, Chromadex, and many more. This is one of the shipments that we received from our vendor. Yeah, and with that many compounds, so we need many freezer to house our compounds throughout our lab. For MS data, we use the Agilent QTOF to acquire the data, and we use AMMS. And for AMMS, we use Bruker timsTOF to acquire the data. And this was our lab on a daily basis. But sometimes things go wrong. Yeah, this happened in July 2017. 10-liter water tank above our lab.
10,000.
10,000 liter water tank above our lab broke and dumped all the water down to our lab. And luckily we have insurance. And sometimes things go right, you know.
After that, the flood helped initiate a significant growth in Medlin and outlet. After the cleanup, many damaged instrument and equipment were replaced. So, and these are many examples of the instrument. And let's take a quick look inside the Medlin database. After you log in, you will see many tabs on the top of the screen, which are used for different types of searches. And here's an example of advanced search. It's pretty straightforward. You can use any information that you have to do the search. In this example, I will use the name search cafe. Once you click search, you will see the result populated. And once you click on Medlin ID, the compound information will display. And if you click on AMMS experimental, the AMMS information of the compound will show up. We collected AMMS data in 4 different collision energy in both positive and negative mode. And if you move cursor over the peaks, you also get the information of that peak. Thank you.
Okay, thanks, Winnie. So now, actually, I'm going to get into a little bit more about the how we've acquired the data, and also how how we're trying to put metabolomics more into the digital biology realm. So Winnie mentioned that we had lots of collaborators on this, lots of ways where we generated these samples or acquired these samples. And one of the key collaborators was an organization that was previous or is still known as Caliber, but Caliber actually became part of Scripps a couple of years ago. And Caliber has a variety of very interesting technologies there, which we've, through our collaboration with them, it allowed us to have that growth spike in melon. So one of their key technologies is this LabSite technology, which is an acoustic deposition device that allows us to acoustically send a pulse of sound energy through the samples from in these 384 well plates and deposit those samples into 384 well plates above. So, I didn't want to. So I'll just go on. I was hoping to be able to stop it, but it's not allowing me to stop it. So those little droplets that you're seeing typically have 5 to 20 millimolar concentration of a particular molecule inside of there. And they're typically on the scale of nanoliter size droplets. So these little droplets that are being shot into the 384 well plate are being deposited into that 384 well plate, which we then dilute further and create a less concentrated version of that particular sample, which allows it to become more mass spec friendly or LCMS friendly. One of the 2 reasons why this is such a useful technology for us is that, one, it's really fast, and the second is it allows us to do multiple sample depositions into one well. So depending on what time we were at, how many samples we were actually getting in, we were able to deposit anywhere from 50 to 100 molecules into each well, dilute them up, send them to the LCMS mass spectrometer or LCMS MS mass spectrometer, and ultimately generate MSMS data for each of these molecules. some of the statistics behind this, and we're really underestimating with this, or I was earlier underestimating the level of effort that Aries and Winnie put into this. So they didn't just analyze 936,000 molecules. They actually analyzed over 1.2 million molecular standards, and with a success rate of about 78%. So that 78% basically means that of each molecule that went in there, of all the molecules that went in there, 22% of the molecules were failing, either because of degradation of the molecules. There's some molecules that perhaps degraded and they just no longer existed, or the signal intensity was too low and it just didn't allow us to get AMMS data. And it's also a probably partly an indicator of Winnie's diligence, because if we had somebody who was less diligent, we probably would have had well over 1 million molecules with AMMS data, but she's actually very cautious about the type of data that goes into the system. So I say this jokingly that if she was less careful, we'd have a bigger database, but because of the carefulness of Winnie, we have a database that is very solid. And I don't want to underestimate the efforts of Aries Ace-Porna. From a bioinformatics perspective, this is a humongous task, and Winnie was able to generate this data, but he was able to play a key role in transferring the information, one from the original lab site deposition-based approaches to the Metlin database, just based on the molecular identities, and also transferring the MSMS data, and each associated with the that molecule. So it was really a huge task on his part, and he really did it extremely well. So each of those 936,000 molecules has MS data at 4 different collision energies in both positive and negative ion mode, and they represent over 350 different chemical classes. And just to give you a perspective, the Metlin is by far the largest database. NIST has grown a little bit since then, but so it's roughly about 5% the size of Metlin, and there are other databases as well, but they're actually quite small. So just briefly, we have grown Metlin into other areas. We have, of course, the fragmentation data, the element composition. We also have an equivalent database that is based on the neutral loss spectrum of all of these molecules, and that's generated from the MS2 data. And finally, we also have an ion mobility database that was recently generated using a Waters SYNAPT2 and also a Bruker timsTOF instrument. And we now have a little under 30,000 molecules with CCS values. Okay, so now you've heard from both Winnie and Lynn about our approach to doing metabolomics or activity metabolomics. Basically, what they do is they generate LC-MS data. They evaluate the LC-MS data for statistical hits, looking for basically a significant amount of fold change, and also evaluating it with respect to p-values for which hits are the most interesting from a statistical perspective, and that's largely done with XCMS. Then we use MSMS for molecular identification. However, this has really turned out to be a major problem, because you've already heard Lynn describe the experiments that she's doing, and she's seeing between 20 and 40,000 peaks. So that means that when she does a typical experiment, and that's just one extraction approach, that's just one chromatography type of an approach, she's seeing an extraordinary number of peaks. And herein lies the problem. What are all those peaks representing? I mean, from a very basic level, we know that some of the peaks are isotopically natural isotopes of those molecules. We know that some of them are sodium adducts, ammonium adducts, potassium adducts, but that in itself can't explain where all these peaks are coming from. And we're generating, in many cases, MSMS data on the vast majority of those peaks. And it's the term associated with this, and it's not a term that I've made up, it's actually a term that's commonly used, and you can see it in papers in Nature and other prominent journals, is the dark metabolome. What are these molecules? So a technology that we actually introduced back in 2008 was similarity searching. So the hope was that, of course, with the vast number of molecules that we have in Metlin, we could do similarity searching, and that could help us identify what this dark metabolome is. And I was actually quite excited about this. This is something that was spearheaded by Paul Benton, who was a part of the staff at that time, and had since gone on to Imperial College and got his PhD there. But it was a really interesting idea on his part, where you could use similarity searching to identify molecules that weren't in the database. So the question is, how accurate is it, and can it be used for identifying what these molecules are? So we've actually published on data in terms of similarity searching since then. The most recent publication was in JASMAS, Journal of the American Society for Mass Spectrometry, in 2022. And this is, I think it's a very telling set of MSMS spectra. So this is a compound, an oxylipin, that was given to us by Jean-Marie Galano out of France, and he's an oxylipid specialist. And you can see the molecules are very similar. You can see this, the only difference between them is this one molecule has a ketone on it, and this molecule has a hydroxyl group. That's the only difference between these two molecules. So if you try to do similarity searching with this approach, based on the MSMS data, it would be a disaster. There's very little similarity between these two molecules, even though they're structurally extremely similar. And dimethylsphingosine and sphingosine C20 are also very similar types of molecules. And again, you can see the MSMS data has very little overlap. Even the neutral loss mass spectral data is very limited in terms of the overlap. Here's somewhat of another extreme example. The molecule on top is rosuvastatin. It's you can see there's a red circle or a red square around the methyl group. The only difference between the molecule on the top and the molecule on the bottom is that there's the molecule on the bottom doesn't have the methyl group on it. Otherwise, it's structurally identical. But of course, you see that the MSMS data is very, very different. I mean, this is one case where the neutral loss data actually was rather useful in terms of showing similarity. But as we've investigated this further and further, we've typically noticed that the MSMS data and the neutral loss data typically don't give you a great deal of overlap between structurally similar molecules. So I'd love to be telling you right now that the technology that we introduced back in 2008 was going to be useful for identifying these molecules, but unfortunately, I'd have to say, based on this and other work that we've done, that similarity searching has been somewhat disappointing in coming up with molecules that structurally identify molecules that are similar. The other approach that, and I have to say, this is something that I've been very excited about. For anybody who knows me, the idea of using artificial intelligence and machine learning is an area which I find absolutely fascinating, and I see it growing in leaps and bounds. So one of the things we did very early on was we tried to use in silico generated identifications based on MSMS data, trying to use that information to populate Metlin with more MSMS data based on what was predicted from the structures of these molecules. And this was, we originally populated Metlin in 2018, and that was mentioned in an analytical chemistry paper back then. However, over the course of roughly 2 years, we realized that the Metlins in silico data was highly erroneous. And this was not only us that was recognizing it, but the really large user base we have. Now we have, between AMMS and Metlin, we have roughly 55,000 registered users, and we're oftentimes getting feedback that the in silico data was just not accurate, not very useful for predicting what the molecule actually was. It was unfortunately, it just wasn't very useful. So as a result, all in silico data was removed, and now Metlin only contains experimental data. So now you're thinking, okay, this is just me just being overly cautious. But recently, there's been some really interesting papers come out from labs that I have a high level of respect for. I think from a computational biology perspective, one of the more I say this in all honesty, one of the brightest individuals in that area in our field is Sebastian Bacher. And this is something that he put out last year, small molecule machine learning. All molecules are wrong. Some may not be useful. So it's a pretty extreme title, but clearly it gives you a sense that at this current time, we're really challenged by using machine learning to identify these molecules. And this year, again, a lab that I highly respect, and probably the most cited lab in the world right now with respect to metabolomics, is David Wishart, and he's a renowned computational chemist. And this paper came out earlier this year. Basically, in a statement in the abstract, they basically say you can't really, there was virtually no overlap between fragmentation ion structure annotations in the MSMS library. So where does that leave us? So that basically leaves us that we can't rely on similarity searching. We can't rely on in silico identifications. So serendipitously, and this is something that you'll recall from last week, we've been actually looking at in-source fragmentation. So in-source fragmentation, and I'm going to go through this relatively quickly because some of you have already heard this before. In-source fragmentation is something that is occurring with electrospray ionization. And the question is, how prevalent is it? Well, we know with electron ionization that's been around for over 70 years that in-source fragmentation is very well known. With respect to electrospray ionization, we didn't know how prevalent it was, and I think until very recently. In fact, a paper that we had in Nature Metabolism a couple of months ago revealed the extent of in-source fragmentation with electrospray. So electrospray is still obviously a wonderful technology. It gives us fantastic molecular ion signals, and as a result, we can oftentimes identify molecules based on their MSMS spectra. However, there is a high prevalence of in-source fragments. And what we did was we looked at the MSMS data in the vast majority of Metlin, over 900,000 molecules we looked at, and we looked at it at 0 electron volts because that mimics the in-source fragmentation that's occurring. So as you may recall, I said that we generate data in Metlin at 4 different collision energies, and it turns out that the 0 electron volts actually mimics in-source fragmentation quite nicely. In fact, the reality is in-source fragmentation generates a little bit more energetic fragmentation than we typically see at 0 electron volts MSMS. So you can see in each of these examples, there's more in-source fragmentation than there is MSMS data 0 electron volts. And based on this analysis of the MSMS data at 0 electron volts, we found that of the peaks that are typically being observed, actually 70% of them are actually in-source fragments. The reality is that since in-source fragmentation is generally more energetic, we suspect that this 70% number is actually fairly conservative. And Science magazine actually described this as these molecules as phantom metabolites. They picked up on this story and they clearly found it as fascinating as I did. So in essence, the dark metabolome is probably largely in-source fragmentation. So where does that leave us with respect to digital biology? Well, I think the answer is we should revisit Winnie and Lin, because Winnie and Lin have always gone about these experiments very carefully in terms of trying to identify molecules that were truly molecules that they could verify existed. And the first question I have to ask is how they've always asked, but we actually asked it in a more specific sort of way this past year. And how reliable is MSMS data? And actually, I said Corey Wang also actually was one of the lead authors on this particular study. So what we did was we looked at MSMS data across 5 different platforms, which it's fortunate that we have these different platforms available to us. We looked at Agilent. Of course, we have the Metlin data from Agilent, but we also looked at the MSMS data that we got from our Bruker instruments, our SciEx, our Waters instrument, our Q Exactive instruments from Thermo, and which one am I missing? No, that's it.
Yeah.
And so this is typical. This is what we were typically observing across the different platforms. So I mean, there's a lot of data here I'm going to show you. You don't suffice it to say that the MSMS data is generally the same, except typically the intensities are going to vary. But overall, you're going to see the same fragment showing up. You don't see dramatic differences, or typically very little differences in terms of the fragments that are being observed. It's typically the intensity of the fragments where you see the differences. And I think that those differences are largely associated with the ionization sources having slightly more energetics than others. And we looked at a wide variety of molecules, and the consistency was generally very good. And here's just an example of the cosine scores that were generated across the instrument platforms. This is all with respect to the Agilent platform, which we were doing a comparison against. And again, here's those same scores looked at it with respect to the number of fragments that were observed in each case. And here's just a quick example of, the MSMS data is nicely consistent across this one particular example of this dipeptide between Agilent and Bruker. And ultimately, the bottom line was that we found, in terms of the best consistency in terms of the ion intensities across those 4 different collision energies, was typically at 20 electron volts. So that's why, at least, that we encourage people to use the 20 electron volts as somewhat of a standard-based approach for doing comparative analyses. Okay, so this is what I'm going to show you right now is unpublished data, and it's the last part of my presentation before we go into Mike Kersey's approach. So we've actually recently integrated XCMS and Metlin. And one of the questions we wanted to ask with respect to this is, how specific can we be with respect to doing these identifications? So I just showed you specific molecules where we have MSMS data for each molecule, and the similarity is quite nice, so that's great. But if we're dealing with what we consider fairly astronomical numbers. 936,000 molecules is a really large number of molecules to actually be analyzing MSMS data against. Are we going to have problems with respect to how many molecules we typically identify when we do this? So we're starting off with, like I said, this huge number. So we did this really simple, and you could almost say it's a thought experiment, but it's really not a thought experiment. It's basically taking the actual data on the elemental compositions of these instruments. We took the precursor ions of these molecules, and we looked at the number of hits across the database that we would observe based on mass and based on the accuracy of the instruments or the errors each instrument has. So at 110 parts per million accuracy, on average, we're seeing about 100 hits. And this is just based on precursor. This is not MSMS data. This is just based on precursor. If we went down to 5 parts per million, which is a more typical cutoff for most instruments these days, we got that down to 50 parts per million. I'm sorry, 50 hits typical. And if we went down to 1 ppm, which again is very doable with current instruments, we were able to get that down to about 10 to 20 hits. So basically, we were going from 936,000 molecules at 5 parts per million accuracy. We could knock that down to roughly 50 molecules. So then we're just relying on MSMS matching of those 50 molecules to do the identification. And that typically got us down to 1 to 2 molecules where you get a really good hit. So I think what I'm about to tell you is very encouraging from a metabolomics perspective in general. So the reality is that MSMS data is very accurate across instrument platforms, and it's very specific. So basically, that high level of accuracy and the high level of specificity across platforms allows us to do really solid identifications across, as long as it's based on standards. It's based on experimental data. So where does that leave us with respect to digital biology? Well, the reality of digital biology is that when you're looking at, let's assume this is a gene network, which it is. I got this from Cytoscape. Each of these nodes represents known molecules, known genes. And this is because the annotation technologies for genes and for transcripts and also for proteins is extremely good, and it's capable to create these highly accurate plot networks that Cytoscape allows us to create. However, with respect to metabolomics, I think this is where we're really lacking, but this is an avenue forward where we, instead of relying on similarity or in silico-based annotation, we strictly rely on MSMS data, whether it's coming from the NIST database, whether it's coming from Metlin or wherever, but we only do annotation or characterization of these molecules based on what I would consider more experimental data, which is from standards, which is going to give you more solid identifications. Okay, well, now it's time for Mike Kurtzey, and actually the timing is perfect. I was hoping to finish this up by 11, so Mike is now going to talk to you about subcellular quantitative imaging of organelles.
Ring. There's a pointer. Okay, great. Thank you. Thank you for inviting me too. So yes, I'm going to talk about subcellular quantification with the nanosims. And so yeah, subcellular quantification. Why? So the tissue is heterogeneous, and yeah, it means that there's lots of different cells which have lots of different functions, and we want to understand those functions in those specific cells. And biochemistry really happens at the subcellular level. So the cell is sort of the fundamental, division of life, but really the pathways for disease and metabolism are really happening at the subcellular level. And so the biggest challenge here is that the cell is an extremely small volume. So Lynn was talking about her analysis, and she mentioned 10 milligrams of material or 1 million cells. So just thinking about it that way, I mean, when you go down to one cell, there's a million times less metabolites or in a lot of our cases, drugs to look at. And then if you decide to subdivide that into, subcellular sections, you're really, you see that you run out of material really fast. So the key is to have, the sensitivity that goes with the high spatial resolution. And so we look at labeled fragments because there's multiple fragments per molecule, and we can put multiple labels on. And so the upshot of this is that you really have to know a lot about your metabolism or your metabolite or your drug before you look for it. And hopefully there's a lot of information in that spatial distribution. And so that's kind of where we're at. And so this is just a schematic of the nanosims and how it works. And so we have an ion source, which generates a primary ion beam. And we focus this really small down to 50 nanometers. And then we raster that beam across the sample's surface. And the sample we use is it's like a TEM section. So it's very, very thin piece of, it's an embedded cell or tissue, which has been prepared for electron microscopy. And so we raster that across the surface, and then we generate secondary ions. And so because we're keeping track of where the primary ion beam is, we can then reconstruct where the secondary ions come from into an image. And then we use a very old-fashioned mass analyzer, a magnetic sector, and this separates our fragments that we're interested in. So typically we look at carbon. We use that as a way to sort of normalize our signal. And then carbon-13 is obviously something that we can label with. This carbon-nitrogen fragment gives a nice outline of the cell. So I think you can tell this is a nucleus. Here's some endoplasmic reticulum. Here's another nucleus. And probably these darker areas here are mitochondria. So we get actually a lot of information from the actual SIMS image itself. And then we can also use sulfur 32 and sulfur 34 as a labeling strategy. We also use halogens, which give a really good signal. And so in this image, we have a cell that was treated with a drug, which was labeled with the bromine label, and then we washed it out after 6 hours, and then we added the same drug, only this time it was labeled with iodine, and we incubated that for 2 hours. And so what I like to say is that the endosome that this drug is trapped in is actually is younger than these, because these have been around for 6 hours. Well, this one's only been taking up material for about 2 hours. And so I said we're using an electron microscopy sample prep. And so that means that before we take the SIMS image, we can actually do electron microscopy imaging to give us a better idea. So you can really see the endoplasmic reticulum here. And yeah, the nucleus is obviously very clear. And then it's fairly straightforward to just overlay these images, and then we get this ultra structural information with our mass spectrum imaging. And you can see that actually I missed this endosome in the TEM, but this interesting structure is quite clear. You can see that the compound is trapped in that structure. And so like I said, we're using this TEM kind of sample prep, and the nanosims is actually more of a material science type of instrument. So it's really designed for looking at dopants in silica for semiconductors and things like that, and also inclusions in geological samples, sort of that sort of thing. But they use this kind of language. They talk about atoms per cubic centimeter, and we said, well, if atoms per cubic centimeter is basically just moles per liter with a little conversion. So we said, okay, so if we can figure out how to take a material science approach and just convert it to a concentration, we may be able to get subcellular quantification at this organelle level. And so, okay, so this is just a little bit about how we make the samples. And so this is showing pellet of cells, but you can do this with tissue as well. And so basically the pellet is spun down, and all the water is replaced with this epoxy, which has a pretty well understood chemical composition. And then I have this cute little square that shows how the cells would look embedded. And then this bullet gets shaved down so that you can cut very tiny sections. I mean, you don't get in a section with one cell, of course, but that's just artistic license. And then we can take that section to the TEM, and that's sort of what it looks like. And so when we look at the sample as a material, we say, okay, essentially there's three parts. There's the epoxy, which remove the water. There's the biomass that sort of displaces the epoxy. And then also we have the drug, or the labeled compound. And so in this example, it's carbon-13. And so the idea was, we wanted to know if we could use the epoxy as you would like to make a standard with a plasma or a blank tissue or something like that. And so we took a closer look at what these things were made of. And so we figured out that the epoxy was mostly carbon, obviously, but it didn't have a lot of nitrogen. And if we looked at what the cell brought, there was quite a bit of nitrogen, but we noticed that the densities were quite different. So then we modeled how much carbon would be displaced, because that would obviously affect our normalization. So if you had a lot of cell material and you tried to normalize against carbon, you could affect you could affect the relative amount based on normalization instead of the location of the actual drug. And so, but what this plot shows is that the percentage of the carbon, whether it's displaced by the biomass, which is 13% nitrogen, or not, it's very stable. And then you can see this in the image. So this is an image of our TEM section. And so here we're measuring the carbon signal. And you can see that even though the cell is embedded in there, you don't see a dramatic drop in the carbon signal. So if the drug is located in this inside this cellular area, and we normalize to it, that we should get a value that's comparable to a blank drug inside of, or metabolite inside of just the pure epoxy. And so this is just the nitrogen image, which shows where that carbon epoxy should be displaced. And we have some graphs showing that it's pretty consistent regardless of the region. And so now that we understand that essentially we can look at the whole sample as a piece of epoxy. And so we know that the ratio of carbon-13 in the epoxy to the carbon-12 should be about 1.1%. And because we know the chemical formula and we know the density of the epoxy, we can calculate that the concentration of carbon in that epoxy is going to be 54 molar. So that means that the concentration of C13 in our epoxy, which we're going to use as a blank sample, so we know that is going to be 0.6 molar. And so in materials world, we don't use percent, we use per mil. So this is just per thousand instead of per 100. So 0 per mil is what we measure in blank epoxy. And so if our labeled drug increases the ratio of the isotopic ratio by 10 per mil or 1%, then we realize that we have 1 micromolar increase in concentration. And so the ideal system would be if we could have a very small spot sort of implanted into a pure epoxy sample, and then that would be the carbon-13 image. This would be the carbon-12 image. And then when we take the ratio, we could see this enrichment in carbon-13. We see this 10 per mil again, which comes out to about 6 millimolar. Sorry, mistake there. So yes, we need to find an ideal system, and luckily we did. And actually, Gary talked about metabolites, which were drugs last time and this time a bit. And I don't know if he mentioned this one, but one of my favorites is the precursor for dopamine. So when you have Parkinson's disease is sort of a degradation of the dopamine containing neurons. And what you do is you give the patient the precursor for dopamine, which is called levodopa. And this molecule is very useful because it can pass the blood-brain barrier, and then it also can enter the cell and then enter the secretory vesicles, which store dopamine, and convert it into dopamine. And so this is a way to increase the amount of dopamine you have in the dopaminergic neurons that are actually still alive. And so this was a good model for us anyway, because this vesicle sits inside a cell, so it's a subcellular structure. It's approximately 200 nanometers, and so the dopamine contains a primary amine, which means that we didn't have to worry about losing it during sample prep. It helps fix with formaldehyde during the TEM sample prep. And so the very cool thing about dopamine is that it's concentrated by this vesicle monoamine transporter, and the concentration is assumed to be constant. And we've seen this in when we incubate cells with L-DOPA, the cells actually swell, and the vesicles actually swell. And so the theory is that the volume is compensated to keep the concentration constant. And there's also a mechanism where this dense core binds some of the dopamine that also allows it to keep the concentration constant. And so our dopamine can accommodate 6 C13 labels. And so 10 per mil equals 1 millimolar. And the concentration can be calculated by comparing electrochemical measurements to the known volume of these structures. So we expect a 600 per mil increase, which we'll show earlier. But so that's the other very, very important part about this model is that we can actually measure this, the concentration inside of the cell beforehand. And so what they do is they take a carbon fiber microelectrode and they insert it into the cell. And those vesicles, which are these little gold dots, so they'll eventually rupture against that electrode. And when they rupture, you apply a pretty low potential, and you can actually strip the 2 electrons off of this molecule. You get 2 electrons per molecule, and that's sort of represented by this peak. And then you can integrate that peak, figure out the charge, and figure out how many molecules are in that structure. And so we had a great idea. So we had a good estimate about the vesicles containing about 60 millimolar, or we expect to see 600 per mil increase. And so we do the measure. So this is a PC12 cell that's been labeled with the C13 dopamine. And you see what happens is we don't actually reach that 600 per mil that we predicted from our known concentration. And so the thing that we realized was that this image is actually a stack of images. And so we're averaging a stack of images. So if you look at each individual ROI, which is what you see here, you see that in this case, these planes are positive for a vesicle containing the label dopamine, but then we've averaged all these planes below which don't have any signal. So then we went through and we looked at each individual structure where the concentration was concentrated, obviously. And I mean, it's also interesting to note these structures are about 200 nanometers, a 200 nanometer sphere. So it's not surprising that they should be about 200 nanometers deep in these sections. And so yes, we look at it, and then we do sort of get to our correct numbers. So you can see that they're all about between 25 and 80 millimolar. And so with that, we had our Rosetta Stone experiment. So now we validated that we can actually read out a concentration from the nanosynth data. And while we were doing this, there was another paper that came out, and they used a drug that contained iodine, and they got very beautiful images of this iodine labeled drug, or it just has iodine in these endosomes, and so rather lysosomes. And we sort of got really excited, thinking, okay, well, maybe we can actually use this better labeling strategy, which gives so much more signal. And so what we did was we synthesized the carbon 13 labeled amiodarone, this drug that contains iodine, and we loaded it into a cell, and then we measured both the C13 concentration, and then we measured the iodine signal. And then from there, we were able to create a calibration curve for the second labeling strategy. And so now we could use this method basically to validate lots of labeling strategies that we would like to use in the future. And so we call this kind of the drug sims workflow. So essentially, we take our labeled drug, we put it into this epoxy that we use for the sample prep. We generate a standard curve like you would in LCMS, and then we do the SIMS analysis, and then we use this calibration curve to convert that SIM signal into a concentration. And that is basically where I'll leave you. So so we can take this materials approach and quantify subcellular uptake of drugs. This material approach can be expanded to different types of labeling strategies, and we can be clever, especially if we know a lot about the metabolism of our molecule, clearly we know that the dopamine would be converted into, or rather, we knew that the L-dopa would be converted into dopamine inside the vesicles. So that was sort of a confirmation that we were looking at dopamine. And the multiple labeling strategies have the ability to track, yeah, well, different parts of molecules. So we've been playing with that a bit at the company, but yeah, we're really excited about it.
All right, thank you, Mike. Actually, Mike told me about this strategy about 6 years ago on a mountain bike trip, and it took me a couple of years to totally comprehend what he was doing. Absolute quantification at the organelle level is really an extraordinary feat. So it sounds like you're even progressing on this even further, Mike, with these
Yeah, I mean, I think
strategies.
it's yeah, I think we've got. I mean, we still have a little ways to go, but I think there's a lot of promise there.
All right, thanks a lot, Mike. And I'm just
Great,
gonna
thanks.
end it with one slide. So we're now done with today's session, and next week it's going to be focused completely on identifying endogenous molecules and that can have biological activity. And as I mentioned at the very beginning, the two speakers, in addition to myself, will be Shu Han from Duke University, who had a recent really nice Nature Protocols paper on identifying biological activity, active molecules from the gut microbiome. And Martin Guerra, as I mentioned, is going to describe this decades-long effort that he's made towards translating endogenous metabolites or a couple of endogenous metabolites into the clinic. So thank you all for attending. It's really encouraging to see how many people are actually showing up to this. We've had over, I guess now 3,400 registrants, and it's really great to see so many people actually showing up to hear these talks. And the last one will be next week. Thank you.